the world of RNA, let us look at another small non-coding RNA, and that is tRNA. So when one talks about tRNA, one always associates it with protein synthesis as it acts as an adapter molecule between the newly synthesized protein and the mRNA. So it is helping in decoding as well as in the synthesis of the proteins. Now, what is interesting to note is that about more than 4,000 tRNAs have been sequenced and studied from 300 different organisms. And this number is only increasing as more and more sequencing of tRNAs are being carried out. And with that, we kind of get an understanding of the tRNA structure and its functions. But there is yet a lot to be understood and associated with different aspects with relation to simple things like why there are certain bases that are modified and how they are useful, etc. Although there is already a lot of understanding with relation to that. So let us look at the learning outcomes of this session. So transfer RNA, which are an integral part of the protein synthesis, have a highly folded structure, and that basically goes from a secondary structure, which is clover leaf structure, to tertiary structure, which is an inverted L. And it has several different characteristics that make it a very significant and specific or distinct identity. These tRNA are versatile in their function as they interact with several different proteins and complexes. So one knows that tRNA can interact with mRNA, it can interact with ribosomes, it can interact with the elongation factors, initiation factors that take part in the process of translation. But there are many more other proteins to which the tRNA interacts and works with. These tRNA get charged. When you say charged, it means they are bound to the amino acid on its acceptor arm, and this charged tRNA, they are able to specifically bind to amino acid tRNA synthetases that make the tRNA charged. So it is considered that every tRNA or every cognate tRNA for an amino acid will have a specific amino acid tRNA synthetase. Now, there are certain specialized tRNA or tRNA-like structures that are reported to have very specific functions during the protein synthesis or in other alternate processes. So, through this session, we are going to look at certain aspects of tRNA, but of course, not everything can be covered in much detail. So the transfer RNA, when you look at the structure of the transfer RNA, when it is synthesized as a linear molecule, but because there are complementary base pairs within the RNA, they tend to form secondary structures, and the end effect of the formation of the secondary structure is that it forms a clover leaf-like structure. Now, essentially, therefore, when it folds into secondary structures, there are very distinct domains that are formed. So you have this domain which is called as the acceptor arm domain. You have this domain which is called as the D domain. This is the anticodon domain, and this is the T domain. Now, within this RNA in this structure, what is observed is there are certain bases or nucleotides which are conserved or semi-conserved. So you will always find these nucleotides at those positions or at those residues, and therefore these have an important role to play in interacting with other molecules and also interacting within itself. So like for example, you can see that CCA over here is highly conserved. You can see that the psi over here, these two G's over here are highly conserved. And because they are highly conserved, it is always observed that these two conserved G can link with C and Psi. And because this affinity towards each other is there, because there is a binding possible between these two, one can see that slowly, because of these interactions that have been shown over here, you can have the secondary structure, which is the clover leaf structure, becoming a tertiary structure. So this D domain interacts further with the T domain to form what is called as the inverted L shape. So you can see how the acceptor arm and the T domain, this forms a particular, a single structure over here, and you can see that D domain and anticodon domain with the variable region present over here form this arm. So now there are two distinct arms present for the tRNA, and this essentially helps the tRNA function as an adapter, because one arm is the acceptor arm that binds to the amino acid, and the other arm is the anticodon arm that has the anticodon that can bind to the mRNA. So therefore, the tRNA is a connect between the mRNA and the amino acid that is going to be in a peptide bond with another amino acid. So that's how it acts like an adapter. What one must understand is that a very distinct feature that is observed in tRNA is the presence of several modified nucleotides. So when you call this as D domain, you're calling this as D domain because there are several dihydrouridine present, and in the T loop there is a ribothymidine and a pseudouridine present. So such modifications or such base modifications are found across all species of tRNA. So they are found in almost all tRNA species. But what has been also observed is that there are certain base modifications which are restricted only to a particular tRNA. So for example, when you consider phenylalanine, the cognate tRNA for phenylalanine, the cognate tRNA for phenylalanine has a very specific modified base called Y-butosine at residue 37. Now you will not find this present in most other tRNA species. So you can see how that certain modified nucleotides make it an identity for a specific tRNA. Now, where you concern or where you look at the structural variability, then what has been observed is, number one, the length of the tRNA can vary anywhere between 65 to 95 nucleotides, and this variation in length is attributed one to the variable region. So the variable loop length can vary from tRNA to tRNA, and second region that is important for structural variability is the D loop per se, and within that the conserved G18 and G19 residues play an important role, because as you can see, this G18 and G19 are associating with the psi and the C. So this is the main point of interaction between the D domain and the T domain. So you can see this D domain is interacting with the T domain, and the D domain is with the anticodon arm, and the T domain is with the acceptor arm. And this is the variable region. The variable region itself is also allowing interaction between one arm and the other arm. Now, what is interesting to note is that three bases can interact in a tRNA molecule. So, say for example, you have A over here. This A is interacting with U. So, you have three bases in an interaction. Say you have um, a bond over here, which is between two nucleotides. You have a third bond formed between another nucleotide. So, therefore, you can see three bases interact. So, it's kind of forming what is called as triples. Okay, so that is very interesting and found in the tRNA per se. Now, again, when you look at the various tRNA molecules, what one has identified with the tRNA is something that is called as structural plasticity, which means that there are changes in their conformation with respect to what it binds to. So say for example, you consider this tRNA and you consider that this is bound to an mRNA. You can see how between the acceptor arm and the anticodon arm, you have an angle of 100 degrees. But say for example, you have the same tRNA interacting with amino acyl tRNA synthetase, then you have the conformation such that the angle between the acceptor arm and the anticodon arm is 65 degrees. So what is indicative of the fact that there is a quite a level of plasticity in the tRNA molecule, but there is also a level of rigidity in the tRNA molecule, and both the plasticity and the rigidity of the tRNA contribute to the final functioning of the tRNA molecule. One another fact that is important for tRNA is that it has non-canonical base pairing. So the fact that it is able to, kind of change its conformation has been attributed to a great extent to the capability of the non-canonical base pairs to become flexible. And so a higher order structure that you observe in tRNA is also attributed to the presence of several non-canonical base pairs. They also have, interestingly, what is called as metal binding regions. And these metal binding regions, in fact, are helping in the folding of RNA. Now, let's say, for example, consider over here that U8 and A14 are in a canonical base pair. So, we know that AU always has a interaction with two hydrogen bonds. But what you can observe is the U is also interacting with another A, but with only a single hydrogen bond. So, this becomes a non-canonical base pair. So, this is something that is again what we are trying to mention, that is present in the tRNA. Secondly, what you can observe is there are very distinct regions where metal ions can bind, and you can see that this is the binding site for magnesium. Now, similarly, when you consider within the tRNA other sets of triple bases together, you will find that they are associated with the magnesium again. Now, the question that comes up over here is, why is it that the RNA molecule has magnesium binding or metal binding regions? The answer to that is that, the basic understanding is that the RNA, because it is made up of ultimately, sugar phosphate present in its backbone, the sugar phosphate is negative, and so the overall negative charge, so when you have the RNA folding, you will have these negative charges coming close to each other, and therefore there can be repulsion. So in order to stabilize two negatively charged backbone close to each other, if there is a metal ion in between, then that metal ion is taking care of the repulsion by acting as a bridge between the two. So therefore, magnesium as or any other metal ion kind of stabilizes the RNA folding. But interestingly, what has been also observed is that if you change the magnesium levels, what is observed that with change in magnesium levels, the folding of the RNA can change. And thereby, that can have implications on the function and the structure both of the tRNA. So therefore, it is interesting to note at this point how metal ions play an important role in folding of RNA. And with respect to tRNA, because it goes from secondary structure to tertiary structure, you will have more of the sugar phosphate backbone coming close to each other, and that interaction is being stabilized by the metal ions. Now we look at the synthesis of transfer RNA. So in prokaryotes, basically, you will find that it is a single RNA polymerase that is synthesizing all the RNA, whether it is mRNA, whether it is tRNA, or whether it is the rRNA. In eukaryotes, however, the tRNA are synthesized by RNA polymerase 3, and when it is synthesized as a pre-tRNA. So this pre-tRNA has to become a functional tRNA, and therefore there is specific RNA processing happening which involves cleavage and base modifications as well. So if you look at the pre-tRNA, you can see how the pre-tRNA is processed to finally give rise to the cloverleaf tRNA. So what are the differences that you observe between the functional tRNA or the mature tRNA and the pre-tRNA? As you can see over here, the 5' end of the tRNA is quite extended with many a sequence of nucleotides being present. So this entire stretch of nucleotides in the RNA is cleaved off by an enzyme which is called as RNase P. This RNase P itself is a ribonucleoprotein, and so it has an RNA molecule which is helping in the catalysis or which is helping in the cleavage. So that is one RNA processing that is done after the pre-tRNA is formed. The second thing that has been observed is that many of the pre-tRNAs have what is called as introns. Just as you have the pre-mRNA having introns, pre-tRNA can also have introns. And these introns can be spliced out, and it is spliced out to finally give rise to the functional anticodon arm. So that is the second processing that happens with respect to the pre-tRNA. The third that happens is that at the 3' OH of the pre-tRNA are present two U's, and these two nucleotides are replaced with CCA by an enzyme system. So you will find that every tRNA will have at its 3' OH the CCA. Other than that, what happens in terms of processing is that many bases are modified. So for example, when you take the D loop, okay, you will find that many U nucleotides are modified into what is called as dihydrouridine. And that is the reason why this is called as the D loop. Similarly, you will find that in the T loop, you have a uridine being changed to ribothymidine, or uridine being changed to a pseudouridine. So you will find several such modifications in the D loop, in the T loop, and also in the anticodon region. These modifications are important or are considered important for the formation of the tertiary structure as just seen before. Once you have the pre-tRNA converted into the functional mature tRNA, only then that tRNA is transported to the cytosol. And the transport of the tRNA to the cytosol is by the exportins that are present, and the exportins take out or export out the tRNA through the nuclear pore complex into the cytosol. Now we look at the processing itself, which means within processing also specifically splicing. So the splicing mechanism in pre-tRNA is very different from that in the pre-mRNA. So we all know that mRNA is spliced out by spliceosome machinery. And in this case, you don't have a spliceosome machinery, but you have a very specific set of proteins that forms a complex that is again removing the introns or excising out the introns. So what's observed in the pre-tRNA is that there is a tetramer in form of a complex which has two endonucleases, and those endonucleases are able to cut at the 3' end of the intron and the 5' end of the intron. So the intron is spliced out. But these two enzymes leave behind at the 5' exon what is called as the 2' 3' cyclic phosphomonoester, and at the 5' end you have the 5' OH being present. Now if you look at having removed the intron, if you look at these two ends, the 3' exon is not able to form a phosphodiester bond with the 5' phosphate, because you don't have a 5' phosphate over here and you don't have a 3' OH over here. So that has to be worked on. So again, there is a multi-step process or a multi-step reaction set, which is ensuring that you will finally have the 5' exon linked to the 3' exon through a phosphodiester linkage. So therefore what observes is that in the process of doing so, there is one kinase and one ligase, and the kinase uses a GTP molecule and the ligase uses an ATP molecule. So what the kinase does is it removes the OH from the 5' end and adds a phosphate. So therefore what happens, or what is present over here, is a 5' phosphate in this exon, and the ligase binds to this phosphate over here. Now, in the meantime, what has also happened is that you have the 2' 3' cyclic phosphomonoester converted to specifically a 2' phosphate and a 3' OH. Now, this 3' OH can actually form a bond with this phosphate. So, the ligase is removed, and now you have the 3' OH attached to the 5' phosphate. So, what is formed over here is the 3' 5' phosphodiester bond. Now the only last thing that has been to be done is this phosphate has to be removed. So that is removed by what is called as the 2' phosphotransferase. And so finally what you have is a mature tRNA with the 5' exon bound to the 3' exon. Now let us look at the transfer RNA making interactions with the tRNA synthetases. So we all know that the tRNA is charged by specific enzymes called amino-acyl tRNA synthetases, and every tRNA has its own specific amino-acyl tRNA synthetase. So what is important to note is there are two classes of amino-acyl tRNA synthetase, that is class 1 and class 2. In this, the example is of cysteine tRNA synthetase and threonine tRNA synthetase. This is a class 2 amino acyl tRNA synthetase, and this is a class 1 amino acyl tRNA synthetase. The amino acyl tRNA synthetase, when you look at its structure, it has very specific domains. So you find that it has an anticodon binding domain, you find there is an acceptor arm binding domain, you find there is a Rossmann fold, which is basically the active site and contains the amino acid after the first step of ADP ribosylation, adenylation. And so uh, you will find that the acceptor arm CCA part is also within the catalytic site. Now, therefore, what is important to note is that the cysteine tRNA synthetase should add a cysteine to the CCA 3' CCA end of the tRNA. What is very clearly observed over here is the way the tRNA changes its conformation when it binds to the amino acyl tRNA synthetase. So, when the class 1 binds to the tRNA, look at the way the anticodon loop is folded, or the conformation of the anticodon loop here is very different from the conformation of the anticodon loop over here. Now, this differences in the conformation or the ability to change the conformation is what we understand by structural plasticity, and that has been attributed to the presence of several non-canonical base pairs and also modified bases. Now the question that comes up over here is, or rather what is to be noted is that the onus of adding the correct amino acid to the cognate tRNA lies with the amino acyl tRNA synthetase. So the question that has to be asked is, how or what is present in the tRNA that tells the amino acyl tRNA synthetase that this is the correct tRNA to which it has to add the amino acid? So several studies have shown that there are very specific nucleosides or bases which actually uh, are giving an identity to a particular tRNA. So if you consider cysteine tRNA, the bases that are modified bases that may be present in this tRNA may be different from that is present in this, some being the same. So for example, dihydrouridine will be same in both. Pseudoiridine will be same in both, but other than that, certain specific modifications or certain specific conserved nucleotides will be specific only for this tRNA or will be specific only for this tRNA. And it is those specific bases that give identity to the tRNA, and that set of nucleotides forms what is called as the identity set for the tRNA to bind to a specific protein, or the protein uses that set of nucleosides to identify one tRNA from the another. So for example, let us say the acceptor arm can have a discriminator base, and this discriminator base allows the tRNA to bind to only a specific cognate tRNA synthetase. So as mentioned, a set of bases or nucleosides can form an identity set for interaction with specific proteins or complexes. Mutations of such identity bases have found effects in its functioning. So when you mutate those specific bases, you will immediately find that the function is changing. Now we come to a very interesting aspect. What we have understood or what we generally associate tRNA with is the protein synthesis system. But what has been observed by various studies, especially in the prokaryotic system, and of course now a lot of study is also being done in the eukaryotic system, is that the tRNA has a role to play in pathways or in systems other than protein synthesis. So one can observe that tRNA can, so when you have tRNA uncharged, uncharged tRNA can have a role in regulation of gene expression. This uncharged tRNA can be fragmented, and these fragments of tRNA can have different roles. You can have this uncharged tRNA having a role in regulation of cell death. So you can see how interestingly an uncharged tRNA being not part of the protein synthesis but being part of various regulatory systems. So under nutritional stress condition, it has been observed that when during nutritional stress the amino acid levels are very less, so therefore the tRNA remains uncharged, and an uncharged tRNA seems to be able to regulate a kinase, and that kinase is able to phosphorylate EIF2. We all know that EIF2 is required for formation of the the translation, the ATS complex for translation to be initiated. Now, if EIF2 gets phosphorylated, it is not able to form a complex with the small subunit, and the so it is not able to form the pre-initiation complex properly. And since it cannot form the pre-initiation complex, translation is going to slow down or it is going to stop. So there is a reduction of global translation, and this is important for survival of the cell or survival of the organism when there is a stress. So that is how the tRNA is helping. Now, another place where it has found to act is in actually formation of cell wall in prokaryotes. We all know that cell wall in prokaryotes have the peptidoglycan layer, and each of the peptidoglycan layer is cross-linked with each other through peptides. So you can have a pentapeptide present. And in some of the bacteria, you have what is called as a pentaglycine. So what has been observed is glycine tRNA glycine serves as a donor for glycine for the formation of the pentaglycine cross-linkage in the cell wall. So you can see how important a role it has in the synthesis of the cell wall, and therefore it is nowhere correlated to the protein synthesis at all. So this tRNA is actually helping in forming the cell wall. The question definitely is, what is directing the tRNA to function for cell wall rather than for protein synthesis, or whether you have a certain set of tRNA that functions for synthesis of cell wall and certain set of tRNA that is taking part in the protein synthesis. Now, another area where a lot of studies have showed the role of tRNA is in the membrane lipids getting acylated by amino acylation. Now, this seems to take place when, say, for example, the bacteria is uh, wanting to resist a pathogenic attack or wanting to resist a stress. So, therefore, again, the tRNA with the amino acid is able to give the amino acid for acylation to the membrane lipid. So, here what you are observing is how a charged tRNA is helping in processes not pertaining to protein synthesis. Of course, its main function has been observed in protein synthesis, but what can also be found is that it is able to help addition of amino acid to certain molecules, or it is able to help in synthesis of, say, for example, tetrapyrrole. Now, another area where it is functioning is in the cell death, regulation of cell death. So what has been observed is that uh, the tRNA is able to add to the N-terminus of a protein, okay, a certain amino acid, and when it adds that amino acid to the protein gets tagged. So the proteasome or the degradosome is able to recognize that protein to degrade the protein. So that is also something that has been observed. So another interesting factor is that the tRNA can be fragmented. So when you look at this pre-tRNA structure, if you have the RNase-P, it is going to remove this part of the tRNA to form what is called as the 5' UTRF. But if you have another endonuclease which cleaves the 3' end, then what you will have or what you'll get is the 3' UTRF. TRF means transfer RNA fragment. So when this is removed, you can again have another fragment present. So let us consider that you can have several different types of fragments being formed from this tRNA. Let's say, for example, take over here. If a dicer cuts at this point, you will have this being released, only this part being released, which is a 3' CCA TRF. If if the dicer cuts over here, you will have this part being released, which is the 5' TRF. If you have an angiogenin cutting at this point, you have got half tRNA being formed. So in mammalian cells, tRNA halves are generated during stress conditions by the action of the nucleus angiogenin. Okay. Now, several studies therefore have shown that these tRNA fragments are associated with regulation of translation during these stress conditions. And it is also responsible for gene silencing. It is responsible for, or they have been found to have a role to play in gene silencing. They have been found to play a role in viral infectivity, and they've also been found to play a role in cell death. Let us look at the example of cell death. So we all know that cell death basically is programmed cell death is what we understand by apoptosis, and within a cell, generally the P53 is suppressed. So when P53 is suppressed and BCL2 is active, then BCL2 is anti-apoptotic. But say for example, P53 is able to elicit the formation of pro-apoptotic molecules. So you have BACs, you have BAC. These will actually create mitochondrial stress in the mitochondria, and the mitochondria will release the cytochrome C from the mitochondria into the cytosol. The cytochrome C, which is now present in the cytosol, can interact with APAF1 to form what is called as apoptosomes. Now, this binding of the cytochrome C to APF1 is what is regulated by tRNA. So how is it regulated? The tRNA can bind to the cytochrome C, and when tRNA binds to cytochrome C cannot bind to APF1. And if cytochrome C cannot bind to APF1, you cannot have an apoptosome formed. And if you don't have an apoptosome formed, you don't have a procaspase acting on it, which will activate further caspase 3, And these caspase 3 will then lead to what is called as apoptosis, that is the death of the cell. So this is very, very clear over here that the tRNA is stopping the apoptotic pathway at this point itself by interacting with cytochrome C. And interestingly, what has been found is that the levels of tRNA are high in cancerous cells, which are transformed cells. So because they are high, they bind to cytochrome C, and because they bind to cytochrome C, those cells, cancer cells, do not undergo apoptosis, and so they will keep dividing, right? So that is something that has been observed in many transformed cells. And also interestingly, what has been observed is overexpression of specific tRNA has been suggested to have a role in regulation of binding to cytochrome C. And therefore, tRNA has been associated with several diseases. Mitochondrial tRNA has been found to be a hotspot for a lot of mutations, and these mutations in the mitochondrial tRNA have been associated with several diseases. Like, for example, very specifically in hearing loss. So, they affect very specific tissue types or cell types. Or it can lead to pigmentary retinopathy or diabetes mellitus or cardiomyopathy. Now, the diseases is not just because of mutations in the mitochondrial tRNA, although many have been associated with mitochondrial tRNA, but the diseases can also be because of misfolded tRNA, or it can be because of overt tRNA expression and abundance, or it can be because of tRNA processing not taking place properly, or because of tRNA modifications being altered, tRNA amino acylation not happening. So all these can lead to various diseases, of which a few are cancers, CNS diseases, that is central nervous system diseases. Many neurodegenerative diseases have been associated with, tRNA structure not being proper. Charcot-Marie tooth disease, this is another disease that is associated with the tRNA per se. So let us make the conclusions. Transfer RNA, which is a homogeneous tertiary structure, has specific characteristics such as base modifications, non-canonical base pairing, and identity sets. The pre-tRNA that is synthesized goes through specific processing to give rise to a functional tRNA, what we can also call as mature tRNA, post which it is charged with its specific amino acids. Other than its canonical role in protein synthesis, it is reported to have roles in gene expression, cell death, cell wall synthesis, viral infectivity, etc. Changes in its specific characteristics and mutations have led to tRNA being associated with several diseases like neurodegenerative diseases, cancers, mitochondrial diseases, etc. Transfer RNA, which is a small non-coding RNA, not just functions as an adapter in protein synthesis, but its structural plasticity, base modifications, and other specific features has attributed it with multiple roles, especially in control during stress. Thank you.